Yes, I think now we are live. Hello, everybody. Welcome back again to this UTS, the Next Genomics educational seminars. Today we have as always, as usual, we have Yvonne Peterman. She's Science and Technology Advisor for 10x Genomics. That she's going to tell us a little bit about the different applications for our theme today, which is cardiovascular diseases and cardiac regeneration. And we have our invited speaker today, which is Dr. Nona Farbehi, that she is from the Garvan Weissman Center for Cellular Genomics. And she's going to tell us about her work. But in the agenda, as usual, we are going to start with Yvonne. Then we'll go straight into Nona's talk, and then we'll have questions at the end for any of the speakers and any of the technical questions or scientific questions for Nona, or whatever you might want to ask. Now, I just reminded that all this technology that we're going to be presenting today, we have it available at UTS. Through the new single cell facility. So again, if you have any questions about how to start your experiment and whether you're interested, you can contact directly. You can contact me. You can contact Yvonne, or anybody on the team from 10x to organize some one-to-one session to guide a little bit on what will be the first steps to do your single cell experiment. But also, if you're an advanced user, that's please let us know as well, because we can cater for any technology really. All right. So with that, any further ado, please welcome Yvonne Peterman, Science and Technology Advisor from 10x Genomics. Yvonne, all over to you.
Thanks, David. Good morning, everyone. Again, also welcome from my end, and thanks for joining today's session. So as David said, I'm going to go and give you a brief overview today over some of the assays and applications that 10x has currently available. I'll also talk a little bit about spatial transcriptomics because it is something that's quite hot and that a lot of people are looking into, and how you can integrate this data. But I will not focus so much on the actual assay that Nona is going to present today, because she'll be going into a lot of detail there, and I really look forward to your talk there, Nona. So as an introduction, which is also how I sort of started all of the other presentations so far, is the three complementary technology platforms that 10x is going to have. At the moment, we have two on the market: the Chromium single cell application with the Chromium controller platform that you see here on the picture. Is sort of the one that we had longest in the market. This instrument, as David said, is available at UTS. It's quite small, and it runs these microfluidic chips, and you can load one of these 4 assay types on the chip and run up to 8 samples if you don't multiplex on these 8-lane microfluidic chips. So as mentioned here, you do put in single cell suspensions, but you can also use nuclei, and that also depends on the assay. So a TechSeq or multi-ohm assay actually only takes a nuclei as an input. And producing viable, high-quality single-cell suspensions is sort of a big part of this workflow, where a lot of the time is usually spent on optimizing this to provide or to generate the best data with from your sample. And the single-cell suspensions can be generated from tissue, from cell culture, from organoids, so from a whole range of starting materials. And then the next platform that we have in the market is our Visium spatial transcriptomics assay. We have recently launched this to be compatible with formalin-fixed and paraffin-embedded tissue sections. So we now have fresh frozen and FFPE compatible with this technology, where you just need a slide, our special Visium a slide, and no additional instrumentation is required to actually read these slides. And obviously you do put in or analyze tissue sections on this particular Visium slide. And what we've got coming next year, and stay tuned for this, is our in situ technology. And you just see a snapshot here where we now allow subcellular detection of RNA molecules directly within the tissue section. So we will provide, more data and more detail as this becomes closer to launch. So, but today it's all about the single cell analysis. So I'm going to now just cover at high level again the end-to-end workflow for generating and analyzing these kinds of data that you can generate on the Chromium controller. So again, here you prepare your cell suspension, and then you load this along with reagent components onto the microfluidic Chromium chips, where we perform the nanoliter droplet generation, encapsulating each individual cell or nuclei, ideally complementing it or capturing it with one of our barcoded beads, and generating these gems or gel bead emulsions. And then there is a number of subsequent steps, library preparation steps that lead you to sequencing ready libraries that are only compatible with Illumina sequences at this point. And within the different applications and assays that we have, and whether or not you decide to run feature barcoding, so putting additional layers on, additional analytes like cell surface proteins or CRISPR analysis, you generate several different types of libraries from your input material within one workflow. And then to complete this end-to-end workflow, we have data analysis and visualization software available for free to download. Cell Ranger in the single cell space, performs your primary and secondary analysis. And with Loop Browser, you can analyze and visualize your data, interact with the data, and generate plots, but you're not actually limited to these analysis softwares. The files, the output files, can also be plugged into a number of third-party tools and that we see frequently used in and described in publications as well. So that's quite an open platform in that regard. This video here highlights how this droplet generation occurs on the microfluidic chip. I just wanted to highlight that, this is obviously carefully balanced with the number of gel beads that you input and the number of cells that you load in to actually optimize the capture and this gem bead generation. One run only takes 18 minutes. So that's quite a fast process. But the one thing to note is that you actually, once you've prepared your cells, you need to be, relatively fast in order to start running it on the microfluidic chip. So having that available at UTS is, from the experimental workflow part, quite crucial. So just some specs on this instrument in general. As I mentioned, you can run up to 8 samples at once, and you can, or what we support is to run 500 up to 10,000 cells per lane. So a total of 80,000 cells you can capture in this with this instrument. We have a fairly low multiplet rate of 0.8% per 1,000 cells, and a fairly high capture efficiency of 65%. Now, what I wanted to mention also is that we have recently released the Chromium X, which is enabling running these chips here and these assays, the standard throughput assays, but it also enables running a new type of chip which enables 16 samples at once, and basically runs or captures twice the amount of cells per lane, so 20,000 cells per lane. So at high level, the 3' gene expression kit here that we've at the top that we've labeled, and that Nona is going to give us some of her great data in a short while. We can capture several things with this assay. So we can at first look, of course, at the whole transcriptome by capturing polyadenylated mRNA molecules of each individual cell that is captured in a single bead. You can leave it at that, or you can add additional layers to it. So you could, on top of capturing the whole transcriptome, also look at cell surface proteins by utilizing antibody mixtures or single antibodies as well, depends on what you're looking at, that have been conjugated with a specific oligonucleotide or specific oligo that is complementary to one of the capture sequences on our gel beads. So that enables us to read these cell surface proteins within each droplet via this linked oligo. So BioLegend has a 10x compatible product range with their total CB antibodies already available that you can purchase or custom put together in terms of specific numbers of antibodies. And we've seen anything from just a few, 5 or 6, to up to almost 100 of these antibodies mixed in one experiment. And on top of this, the 3' gene expression solution also enables detection of single guide RNAs. You can do so you can perform CRISPR perturbation experiments where you capture or measure the actual guide RNA within the cell. You measure the effect this guide RNA has on the transcriptome because you capture that at the same time. And you can basically match these effects of the guide and the perturbation it sort of affects in the specific cell. Also, the workflow for this CRISPR screening is a lot shorter than some of the sort of traditional CRISPR assays that you can run. Just keep in mind, though, that in terms of the throughput and the number of guides that you can test, is in a way limited by the number of cells that we can capture per lane, which is 10,000. So you can typically do up to 40, 45 guide RNA screen or tests with one Chromium controller run. And how this is enabled, just wanted to quickly here show at high level, is the different types of oligos that we have on these gel beads. Again, at the top here, our poly-DT oligo captures the polyadenylated mRNA, but with capture sequence 1 and capture sequence 2 that we've introduced to these beads, that allows us to look at the cell surface protein as well as the guide RNAs, but it also allows our cell multiplexing, which we've introduced with our CellPlex kit. And you see this here at the bottom. It's a lipid tag based labeling method. So the cell multiplexing oligo integrates into the cell membrane or nuclear membrane for labeling. So prior to pooling your sample, you would incubate your sample with these CMO tags and perform multiplexing in this way. So at high level, the workflow is shown here. You can input cells or nuclei, although it is recommended to do this on fresh samples and not frozen only. And typically we see, in terms of handling, pooling 3 to 4 samples together seems to be where the sweet spot is for the CellPlex product. And in the workflow, as mentioned, once you add in another layer of analyte, you will basically generate a separate library to capture the cell multiplexing information on top of the CRISPR or cell surface protein information. And also just wanted to highlight that for each of these types of libraries, we recommend a certain sequencing depth. Here for gene expression as a guide, it's 20,000 raw read pairs per cell. And for any of the feature barcode libraries, it's less, it's about 5,000 read pairs per cell. And maybe Nona can comment on that in her talk, whether she thinks that that is sufficient, or whether in a lot of cases people do decide to go a lot deeper with their sequencing than our recommendation. So again, this is within our software, we have this demultiplexing of samples also inbuilt, so you don't need to create any software or any scripts to do this. It's all already incorporated. Now, the next assay that we have available is our single-cell immune profiling assay, or we call it 5' gene expression as well. So typically here we also can look at the transcriptome-wide gene expression, but we are looking actually here at the 5' end of the transcripts, and that allows us to capture full-length paired VDJ sequences from T-cell and B-cell receptors. So this allows you to capture a lot more detail than going in from the 3' gene expression, although even there we see actually in the literature very effective detection of immune cells, for example, just based on 3' gene expression. But in the same solution here, we also have enabled feature barcoding, where you can add another layer on top, again, cell surface protein detection, with BioLegend's total seq-C product range, similar process here or mechanism via these little oligo tags that are added on. And we can also use or look at antigen recognition or antigen interaction for T-cell and B-cell receptors with the immune profiling solution. Cells, here. It wasn't advancing, sorry about that. So to show you what this kind of data would give you is highlighted here in a data set that we've generated in-house on PBMCs that we've incubated with the total seq-C antibody panel from BioLegend, with detecting 17 different cell surface proteins. And then we have run this through the Chromium controller and generated the gene expression immune repertoire profiling library and also the feature barcode as cell surface protein information. And here we are looking at the t-SNE plots that are being generated, looking at the cell types that have been annotated using this 5' gene expression information from the kit. So you can see, we capture and identify a large number of different immune cells within the data set. But when we add in the gene expression, when we add in the cell surface protein information from the antibody set that we've incubated the cells with, you can actually see that now the annotation becomes improved. So you see this clusters are being more distinct in the data set than just with gene expression alone. So you can basically increase the resolution of your data set by using these total seq antibodies. So this is an example of what it looks like if we do capture a T-cell receptor sequence of a specific cell in the assay, and if we do have this, we can get specific clonotype information for the T-cell receptor. So here in the example, the blue cluster, but also in the sorry, did I skip a slide? Sorry. In the T-cell receptor here in this panel, we can see the full-length sequence as well as the clonotypes, and we get the same information for the immune globulins, full-length sequence as well as clonotype information. And as an example of this implementing or utilizing this assay is this publication here. Published in Stem Cell, or in Cell Stem Cell in 2019, they actually utilized both assays, the 3' and 5' gene expression solution. And the authors here took neonatal mouse heart cells, and they chopped them and plated them, grew them up, and then they infected these cells with lentiviral cassettes that impact three key genes, transcription factors of cardiomyocyte development and fate determination. Then they performed the media swap and induced the cells to differentiate towards cardiomyocyte lineage, and then harvested them at 3 different time points. And this is then what they subjected to the Chromium controller run. So they performed 3' gene expression, but also analyzed with the 5' run here, with the immune profiling run, in addition with the cell types, and were able to deconvolute different stages of cardiomyocyte differentiation. So really interesting example of actually combining both of these assays in cardiac research. So next up is the single cell attack, or the combined ATAC-seq plus 3' gene expression, or what we call the multi-omic kit. So ATAC here is our version of looking at the epigenome in every single cell. So it's the assay for transposase accessible chromatin. So we are looking here at accessible chromatin structures, and we capture that as fragments that get tagged with a specific oligo. And at the same time, with this combined assay, we are also detecting the transcriptome of each individual cell. But as I mentioned, originally, we are actually inputting nuclei in the solution. And Nona is actually going to talk in December about her experience with this kit, which I'm also quite excited about, and we'll keep you updated with further details. And she will also mention, you know, some tips and tricks around optimizing nuclei extraction to perform an assay like this. And if you make it work, you know, if you can capture the gene expression data at the same time as the accessible chromatin, you know, it allows you to characterize these gene regulatory networks and cell states in your sample. And you are able to directly link the gene expression and the specific attack modalities cell by cell or nuclei by nuclei. So you get this direct linkage between each captured nuclei, and you can, in the analysis, you know, obviously look at gene expression alone and the attack chromatin fragments alone, but you obviously can also match this together with a specific separate Cell Ranger ARC tool that we have available to perform this analysis. So here, this is a snapshot of what this looks like, specifically when we look at the LEF1 here at the top listed. We look at the global link for LEF1 and indicated in the open chromatin peaks. They are either correlated, and that's highlighted in blue at the top, or anti-correlated, which is represented by these red arcs, with the LEF1 gene expression across the 1 megabase window for the same nuclei that we've just seen on the last slide. And we can see here that also that the LEF1 expression levels and open chromatin peaks, they're colored in color-coded by cell type, and you can see cell type specific expression of LEF1, and that's correlated with these linked open chromatins. So just an example of what this kind of data looks like and how you can zoom in on specific areas of interest, genes of interest, and the kind of information that you get from this if you're looking at Loop Browser in this case. An example of where this has been put into action is this one here, where Alexanian et al. Just recently published this in Nature, where they're looking at heart or fibroblasts, and they identified, you know, a transcriptional switch that governs fibroblast activation and heart disease. So they've actually done the assay separately, attack and gene expression separately, and were able to, you know, shed light on a specific activation and chromatin accessibility that they were previously not able to describe in this kind of data set. Also wanted to cis mention here that they've also utilized CRISPR interference to look at specifically 3 controlling as cis element controlling MEOX1 promoters, and they were able to then again further fine-tune the results that they've achieved also with a single cell data set. So there is a lot more hidden in this paper, and I just wanted to highlight these 2 things from this publication. So if you're interested in looking into this in further detail, please let me know. And I know we are sort of on the 20-minute mark now. I just wanted to, with the last few minutes, quickly highlight the Visium Spatial Gene Expression Solution. that we have and how it works, or how this could potentially be applied in the cardiac research area. And here's an example from cancer, from a cancer sample that's left. It's H&E stained, and on the right we are looking at tumor infiltrating lymphocytes, where we've tracked where they occur, and at the top here they have infiltrated, providing a good prognosis for this particular patient. And at the bottom, again, we are looking at a tumor section here, H&E stained, and the infiltrating lymphocytes are being held back, which relates to poor prognosis for this patient. So being able to track this spatially with the gene expression really tremendously helps with diagnostics, and also potentially identify mechanisms of how this works in specific cancers, and how to potentially tackle improve treatment. You get two different types of data from the spatial gene expression. So you capture the histology image and then the gene expression, which here in this case, each dot represents one of these barcoded spots on the slide. And then you can overlay this and can see where these different expression patterns occur in the tissue. For Visium, we also have enabled at the same time to look at protein expression and capturing gene expression of the same section on the same slide. At the moment, it is limited to 6 different antibodies that you can apply, and this is an example of human breast ductal carcinoma immunofluorescence data, as well as overlay gene expression. The workflow is again also ready to use and robust. It can be, in the case of fresh frozen, completed in 1 day. For FFPE, it stretches out to 2 days, because we are incubating probe pool on the slide overnight. And again, it comes with the data analysis visualization tools. And as mentioned, you actually don't require a specific instrument, but you will need a tissue sectioning capability in your lab, so like typical histology equipment. And at high level here for fresh frozen, it is actually species agnostic technically. So we have customers also that are utilizing this on plant species, for example. And for FFPE, it's currently enabled for human and mouse, targeting 18,000 or 20,000 protein coding genes respectively. The resolution of the solution at this point here is highlighted in this slide, where the 5,000 barcoded spots that we find within these 6.5 by 6.5 millimeter capture areas have a diameter of 55 micrometers and center to center a distance of 100 micrometers. So that sort of gives us a resolution of 1 to 10, and in some cases to 20 cells, depending on the size of the cells that you're working with. So typically what we see is that customers are performing a deconvolution analysis to bring the data set down to the more single cell resolution. And then hopefully, in the first half of next year, we will have Visium HD launched, which will enable the resolution, or which increases the resolution of Visium spatial gene expression down to single cell or sub-single cellular level, with these barcoded spots being 5 micron in diameter. There's also a whole range of other launches coming up that will enable and make this Visium workflow much easier. And this here is a snapshot of the increased resolution that we are expecting with the HD coming up. And I think in the interest of time, I will skip the integration of utilizing, these data sets, Chromium single cell data set and Visium, and increasing them. And I might just finish off with letting you know that we have a virtual user group meeting coming up that's going over 2 days. And it will, if we can make it on day 2, for example, our technical team here will talk about some of the ins and outs of that single cell and spatial data generation, sample prep details, etc., to give you really good insights into, getting started with your experiments. If you can make it, please feel free to register. That's coming up on the 17th and 18th of November. And out of this here, if you want to discuss anything in further detail that what you've seen today at high level, you can get in touch with us and the Science and Technology Advisor team and Field Application Scientist team here in Australia. We are more than happy to meet with you and have an in-depth discussion about a specific project that you might have in mind. So with that, thank you very much for your attention, and I'll hand back to you, David.
Well, thank you very much, Yvonne. I hope you can hear me. Unlike last time, my microphone stopped working. So thank you very much for your perspective on the different technologies that 10x can offer. I think that they're increasing by the day. So this is a wide variety of things that you can do, single cell and high resolution. So now we have our invited speaker, Dr. Nona Farbehi. She is currently a postdoctoral researcher with Professor Joseph Powell's lab at the Garvan-Westman Center for Cellular Genomics. And she did the PhD with Professor Richard Harvey and Robert Nordon from UNSW. So she's coming from a very strong team of cardiac research. She is using large-scale single-cell transcriptomic analysis of cardiac stromal and stem cells, and now she is also doing some induced pluripotent stem cell analysis from patients and differentiating them towards different phenotypes and understanding how that dynamic interacts with the DNA. So today, Nona is going to talk us about insights on cardiac biology using 10x Chromium technology. Thank you, Nona, for being here today.
Hi, everyone, and thanks, David, for the introduction. It's my pleasure. And thanks, Yvonne, for the great presentation on various aspects of 10x Chromium technology. So I might start sharing my screen. Hopefully you can see it well.
All good, yep.
Okay, nice. So today I'm going through some project that I have done during the past 4 years, focusing on cardiac biology, and most of this part is including 10x Chromium technologies, various platforms. As you may know, going through the cardiovascular disease, so it's the #1 killer all around the world, and one of the most prevalent cause of the death is heart failure, which is most of the time is following myocardial infarction, which I refer to MI from now on. And one way to tackle heart failure is to look for the ways to either replace the cardiomyocyte regeneration in the scar region, in the part of the heart that has dead cells after the infarct, or try to repair the heart through various techniques. For example, if you can see in this figure, various researchers employ different methods. For example, trying to induce proliferation in resident cardiomyocyte after the heart attack, or using bone marrow stem cells to regenerate the heart in the infarct region, or using the resident cardiac stem cell and targeting those to regenerate in the scarring part of the heart. So in which we call them cell-based therapies. And then various studies prove that it's working. Some are in trial still in big animals and in a human, but still a long way to go. So, so far, our lab is focusing, like my previous lab, Richard Harvey's lab, focusing on a portion of cardiac progenitor stem cells. And then various labs look for different markers to enrich for those cells, like c-KIT positive cells or SCA1 positive cells, IL-1 positive. So our lab was focusing on a subset of these cells using two main markers, PDGF receptor alpha positive and stem cell antigen 1 positive, and excluding all the other endothelial cells or immune cells. So we call them cardiac, so in vivo we call them S plus P plus, which refer to PDGF receptor alpha and SCA1. And in vitro, we call them cardiac colony-forming unique fibroblasts. And I will explain for you why. And the main reason I'm focusing on those cells is activating the endogenous CPCs, those cardiac progenitor cells, will form new smooth muscle cells, endothelial cell, and also activate the fibroblasts, which help the heart modulate the extracellular matrix and promote neovascularization in the part that has like dead cells, scarring part, and which resulted in enhanced perfusion and improved cardiac function. So the goal here is to try to reduce the scar region and maintain the ejection fraction of the heart to make sure the heart failure is not happening after occurrence of MI. So in this study, my goal was to uncover the transcription profile of those CPSC in steady state and also in disease, and look for molecular mechanism underpinning the reparative effect of these cells, which weren't completely understood. So in the first set of experiment, we have induced MI by ligating LAD, which is artery going on top of the left ventricle. In this figure, you can see two hearts. The heart on the right is like a sham heart, it's like a healthy heart. And the heart on the left part is the one that we ligate LAD with a suture, like a black suture, and you can see scarring on the left ventricle. Then furthermore, and this is like a wild type black 6 mice. And then we cut the ventricle, and then we dissociate the cells and we push them through the 10x chromium 3' channels. So as Yvonne explained earlier, the cells and the beads is going to capture into the like micro drops, and some drops has like one bead and one cell, some drop has only the bead. And the efficiency of this kit is quite high compared to other droplet-based methods. So more than 60% of the droplets will have both cell and the bead. And the bead has like poly T ends, which absorb all the RNA, mRNA from the cells in those drops. And furthermore, we did the library prep of these ones, and we will see the data. So this is how the data looks like. So we will use, most of the time we are using TSNA plot or UMAP plot to visualize the data. Each dot represents a cell. And here on the top left-hand side, you will see the sham hearts. So in the normal healthy sham hearts, you will see a majority of the fibroblasts and endothelial cell, and also like B cell, T cell, the immune cell subset, and a very like major cell type there as a resident cell. So, and on MI day 3, you will see like a flux of monocyte macrophages in the tissue because of all of those inflammatory response of the heart, and you capture less of those fibroblasts and the endothelial cells. MI day 7, you see an appearance of different type of immune cells. That's the green population, and also the blue population of myofibroblasts. So we have shown like this is the aggregate of tip cells on the lower left. We have presented like a very high resolution of various cell type residing in the heart after MI, 3 and 7 days after MI, and also in the sham heart. And we can see like we capture various type of fibroblasts. So like those ones who are expressing SCA at high level and SCA low level, which we refer them as FSH and FSL, and also activated fibroblasts. The cells start to showing activation marker like periostin, and myofibroblasts are start to appear after like MI to deposit like extracellular matrix and collagen in the site of the injury to maintain the structure of the heart. If you don't see the fibroblast formation, then probably you will end up in cardiac rupture and death. So this is we like myofibroblasts. So, and we see a lot of various endothelial cell types residing in the heart, motor cells, the cells in cycle, and various type of macrophages, monocyte, M1, M2, dendritic cells, and B cells, NK cells, and glial cells. So here in this plot, I'm showing you the cell type marker genes like collagen 1 for fibroblasts, periostin for those marks for those activated fibroblasts, KDR for endothelial cells, and also a really tiny population of glial cells marked by PLP1. And we then propose a statistical test called differential proportion analysis, which is a kind of permutation-based test that we look at how the cell number varies in between that different time points and whether these differences is significant. And we've seen like the differences in number of M1 macrophages are very, very significant from day 0 to day 3. And also the same for endothelial cell, we have a decrease from day 0 to day 3 and another increase here. And furthermore, we've seen a shift from pro-inflammatory type 1 macrophages to pro-reparative type 2 macrophage M2 shift from day 3 to day 7. This is really important because when a disease and something like MI happen in the heart, you will see infiltration of Ly6C high macrophages from bone or spleen to the injury site, and they differentiate to Ly6C low monocyte macrophages, and then they start secreting various cytokines. So at day 3, you will see like a lot of TNF alpha pro-inflammatory cytokine, and at day 7, which is a healing phase, you will see a lot of anti-inflammatory mediators depositing in the site of the injury. So the balance between these two types are really important, because if this balance, if there is a like blocking, if you block the inflammation at day 3, you will get cardiac rupture. And when you get excessive inflammation at day 7, you will end up with a dilation and adverse remodeling. So early resolution of inflammation give you like a smaller scar and better vascularization. So this data set in single-cell RNA-seq help us to unravel this gene expression dynamic of macrophages during like inflammation and healing phase following MI. And then to be able to tease out different type of monocyte macrophages, we look at the marker genes indicating like expressing solely on those clusters. And you see here in the dot plot map that for each individual cluster, we have at least 4 or 5 individual markers that is explicitly expressing in those clusters. And when you look at the biological process term, you can find the relevant term in regards to each individual subset that we named them. For example, we found interferon inducing cells in like the pink one, macrophages, and all the biological term came up was about the response to interferon beta, gamma, and alpha here. So this map gives us like a thorough transcription profile of the various type of monocyte macrophages residing in the disease heart. And then we built a very thorough ligand receptor interaction map by utilizing a data set of human ligand receptor pairs, which is published in Nature.com in 2014. And we develop a list of mouse autologous comprising of more than 1,000 ligand receptor pair. And in this figure, you can see like the whole map. And for example, the myofibroblasts in blue, is talking through ligand receptor pairs with themselves and also with various type of cells around them. We don't have the special data for this crosstalk, but then using this map, we can go back and check our like IF staining to see whether you see any cells like any myofibroblasts in the close contact like to like F skylo or other cell types, and we did that. And when you look at the significant incoming outcoming connection, you see that myofibroblast has like a highest outgoing rate. I mean, and this some cells are more receiving, some cells are more like transmitting the signal, and myofibroblasts are like more of transmitter. And then we have like EC1, the endothelial cells that have more incoming signals. And looking at, for example, glial cells, we can look for the triplet of outbound connection, like to see which ligand receptor pairs glial cells are employing to talk with endothelial cells or mural cells. Furthermore, in a recent paper, which is more like a review paper, we focus on PDGF ligand A and B, and then how they crosstalk with how different cell types are using PDGF A and B ligand to talk to other cell types through PDGF receptor alpha and beta receptor. This is just published like a few months ago, and we also look at the staining of PDGF receptor beta and see, for example, here around the vessels, you see a lot of PDGF receptor alpha GFP. And then when you are looking at the crosstalk map, you see the same thing, that when they reside in the close proximity in your sections, and then you see from the ligand receptor map, you see like the crosstalk as well. That's really reassuring. So furthermore, we look at diffusion map of day 3 and day 7, which give you an idea like how the cells differentiating during the time. For example, we see this like a diffusion map shows us the unactivated fibroblasts going through differentiation toward myofibroblasts and toward the cycling cells, and partially toward the Wnt cells. So the Wnt cells is a very interesting population because they're expressing a lot of Wnt related genes, which I will explain like shortly. And this is like segregation of the diffusion map based on various days, like sham, MI day 3, and MI day 7. And that's where you see the differentiation from unactivated cells toward myofibroblasts. And in the heat map of differentially expressed genes between clusters, you will see relevant biological term in regards to, for example, the cell cycle genes versus other cell type. The terms is solely about, they are solely about cell cycle, which is confirming our observation. Furthermore, using, so by focusing on just a population of this plus P plus cells, we have done more single cell RNA-seq. So the reason was previously in the publication in Richard Harvey's lab in Cell Stem Cell 2011, the lab shows that by purifying the SCA1 positive cells and PDGF receptor alpha positive CD31 negative, and for enriching for those population and putting them in the dish, you will get various colony forming unit fibroblasts, which we can maintain in vitro for so long. I mean, mainly the large population, large colonies, and they are kind of stem cell like cells in vitro, but they are quiescent in vivo. So no division in vivo, but they start dividing in vitro. And they can give rise to, we can differentiate them to various cell types like smooth muscle endothelial cells. So by focusing on just a portion of these cells, like PDGF receptor alpha positive CD31 negative using a transgenic mice, we want to look for whether we can get a better overview of these cells and whether we can find like therapeutic opportunities for targeting these and to be able to restrict the reactive fibrosis. So this is the overview of the data, the second data set that I'm talking about. So we capture GFP positive CD31 single cell RNA-seq, and you can see that there is a lot of clusters showing up, and then we even capture a tiny clusters of epicardial cell at day. Epicardial are in like green here at day 7, which is really interesting because in the literature we have seen that after myocardial infarction, some cells revert back to their developmental origin and start expressing epicardial genes. And then we capture a very higher resolution of various types of fibroblasts within the murine hearts. And the data really is confirming with the previous data set that I explained for you. We call the initial data set the TIP cells, the total interstitial population. And these one, the GFP cells. So we did the population percentage. We look at the population percentage, and we figure out at day 7, we have a lot of myofibroblasts within the cell cohort, and this SCA low population is kind of diminishing. And at day 3, we have a peak of appearance in activated fibroblasts, which is quite interesting. And also the Wnt-X population is kind of increase and decrease at day 3 and day 7. And then this is the plot showing the marker genes across GFP population. So all of them are positive for collagen 1, but only a proportion are expressing Ly6A, which is the SCA1. And like all of them are, majority of them are positive for PDG receptor alpha and also the GFP protein. And then this is the top marker genes across the GFP population, which you can find in the publication, my publication as well, in great detail. So by focusing on the population that is expressing the Wnt-related signals, we found that most of the genes that are expressing are kind of down-regulating Wnt canonical and Wnt calcium signaling pathway. I color them in red, so like SFPR and VIF1, so there are Wnt inhibiting factors, and there are like other proteins involved which are upregulated in the sense that they have negatively regulated the Wnt signaling. And then when we look at the ligand receptor pairs, we realize that these clusters are mainly talking with endothelial cells through various ligand receptor pairs. And I just highlighted here that Wnt-X are these population, and the endothelial cells are the one on the low side. And these are the markers, like main markers here. And by looking at the biological process term, you will see exactly the same thing, like negative regulation of Wnt signaling. And we thought, so it's worth investigating more. Then we did some immunohistochemistry by looking at the protein expression of the VIF1. And it was a bit tricky because VIF1 protein, it's not really well studied. So we did a fair bit of experiment to be able to set it up in the lab. And then we found that in cardiac cells, VIF1 staining is perinuclear, and we demonstrated co-expression of VIF1 with a Golgi marker here, GM130, which is consistent with VIF1 being a secreted protein. So the location of the expression was correct. And we also see a few cells. So in the sham part, it was really hard to see any VIF1 positive cells. At MI day 3, we've seen a few of them, like I have the quantification here. And then a few of those we found positive cells at MI day 3 were actually in cycle using Ki-67 marker. And there were a very few cells that were both GFP positive and VIF1 positive, as we've seen in the single cell RNA-seq data, and also very few cells which were CD45 positive and VIF1 positive. So as per our predicted cell-cell ligand receptor connection, we've seen the same in our tissue, in our sections, which is reassuring. And based on the single cell RNA-seq data, we built this map, which is a schematic summary of the differentiation dynamic of GFP population. So for example, in the sham heart, we've seen that fibroblast SCA high can give rise to themselves and also to the activated fibroblast. In MI day 3, FSL SCA low is diminishing and FSA high, SCA high is also diminishing, but the activated one fibroblasts are increasing actually. On MI day 3, you see like a decrease in the number of FSL and FSH, but decrease in the F trans, which is a transition time, fibroblast in transition, and then also VIN6, and also those fibroblasts in cycle. And there is like these dots, they are like, we think they might give rise to each other, but we are not sure. It requires further investigation. And then in another study that is published just today in Cell Stem Cell, I was involved in the single cell part of this study. So Vaibaho was the main investigator together with Richard. So we look at the cardiac fibroblasts and we found that they are more hypoxic than other cardiac interstitial population and express more HIF1 alpha and exhibit increased glycolytic metabolism. And why to tell this? Because when Vaibaho was looking at the hypoxia index of those cells, S plus P plus cells, which we call them cardiac fibroblasts, he realized that there is more staining for pimonidazole. And also when you look at glycolytic flux, also there are like HIPs more in S plus P plus. And the same for mean mitochondrial mass. He used like a mitotracker dye, and they were lower in S plus P plus cells than like the total interstitial population. And when he looked at different portion of the mitotracker dye, like just sorting for low and medium and high mitotracker cells, which is colored in various like blue and green and red, and put them in cell culture dishes, he realized that only the majority of the cells in the low proportion will give rise to the colony forming cells, which suggests that hypoxia is a preferred environment for the cardiac colony forming cells with long-term proliferation capacity. And then to explore what's happening, Vaibaho generated like cardiac fibroblast specific HIF1 alpha knockouts mice by specific breeding, like PDG receptor alpha mercury driver mice with HIF1 alpha flux mice. And I did perform single cell RNA-seq on TD tomato CD35 minus CD45 minus cardiac fibroblasts. And this is how the data look like. We have seen that this is the experimental setting when you induce, when you inject tamoxifen at 8 weeks, and at 10 weeks old, you do the MI, and then you start analyzing at various days. This is at day 3 post-MI, this data, this single cell RNA-seq data. And then we figure out the cardiac fibroblast specific deletion of HIF1 alpha resulted in decreased HIF1 target genes expression and actually increased mesenchymal progenitors in uninjured hearts. And then increased cardiac fibroblast activation without proliferation following sham injury, as demonstrated by these single cell RNA-seq data. So and also losing.
Nona, sorry to interrupt. We're running out of time. It's already 11. So if you can wrap up, thank you.
Sure. Thanks so much. So I might skip through this slide. So that the paper will be live today. So it's better to have a look. And then this is my current project. I'm using an iPSC drive from individuals reprogrammed to cardiac cells and running on cardiac like single cell multi-ohm kit from 10x in parallel to genotype data and function analysis to look for genetic effects on cell fate at population scale. And as a conclusion, we characterize a major non-myocyte cell population, build a ligand receptor network, and provide a resource of various cell type in cardiac disease and also in health. And I would like to thank all the people involved, specifically Ralph here, and any questions. So,
thank you, Nona. Sorry, everybody, we're going a little bit over time, but we do have a question. So from Lana McClements, and she was asking when you were presenting that data before the last paper, but in your previous live paper. So what is the significance of increase? What is it? This is periostin expression concomitantly with reduced collagen 1A1 expression in terms of cardiac health and fibrosis?
So that's a good question. As far as I remember, the periostin was expressed higher in activated fibroblasts. But I can't recall whether there was a reduction in collagen 1A1 expression. But there is a nice paper from Wolfken team on periostin, on a mouse model, looking at the flux and the head model for periostin, and exactly explaining what she is asking. I can share it with you. I can't recall what was the result from that paper, but I can share it with you. Yeah.
Yes, Lana. I don't know if you see this, the Q&A, but Lana says yes, please.
Yes, I will share it.
And then just in the interest of time, I'm just going to go one quick question that I've been sort of thinking all along through your presentation. And it's like, you know, you're showing all the cell-to-cell interactions between endothelial cells, fibroblasts, cardiomyocytes, and all these sort of immune cells. So what do you think about trying to recreate all these connections into organoids? And how do you think that, for example, you know, that field of therapy will, you know, be able to at some point recreate all the complexity that you can see in a, you know, like when there is a wound healing, sort of a regeneration sort of after injury? How do you think that field going?
That's an excellent point, David. If we can find a way to do it in vivo setting, that would be awesome, like looking at organoid and how the cells interact within those organoids. And you're the expert in that field, so I probably leave it with you. But at the moment, because my previous lab didn't have any expertise in the organoid models, what they did is they look at 10x, like the Spatial technology, and did the sectioning and tried to map it back to the single cell, the ligand receptor pairs that we have the data on. So that's what Ralph is working on at the moment, following up that experiment. But it would be great to have like various set of data set, like in vivo, in vitro, like organoid model, as well as like actual sectioning of the cardiac tissues, and try to see whether you can actually see what's happening in the body, like how and what's the goal of these interaction, like.
Yeah, thank you very much. I think that's it's a good point. I mean, it's the combination of different data sets in the end what is going to give you, you know, a much clearer answer of what's going on.
Exactly.
Well, thank you very much. I would like to thank you for coming today and presenting your beautiful data and showing with us. And also thank Yvonne, as always, for your presentation. And I'd like to just letting you know that in the next session, in 2 weeks' time, we're going to have Alain Faith from UTS, and the topic is going to be in respiratory diseases. So I hope to see you all there. And yeah, I'll see you next week. Thank you very much, everybody.
Thank you. Thank you for having me. Thanks so much, everybody.
Thanks, everyone. And congratulations on the publication today, Nona.
Thanks.
On the publication of it.
Yes, congratulations.
Yes.
Thank you.
Bye-bye.
Cheers.
All right,
Bye.
cheers. Bye-bye.